So good morning, greetings to you all. This is teacher Dr. Mgombo Ronald. We are back again. Today we are going to go ahead and look at the central dogma and transcription. Now, so if you are new here for the very first time, consider to like the YouTube channel, consider to share, consider to subscribe, because that is the only way you can support us, that we can be able to grow, encourage us to go ahead and make more videos for you, to make biology a little bit simpler for you to understand. So today we are going to go ahead and look at the central dogma and transcription. So when you look at the central dogma is simply, it was coined by a gentleman known as F.H.C. Crick in 1929. When he was coming up with the central dogma, he said this is some kind of flow of information, how information flows in a cell. How information flows in a cell from the DNA, which is usually replicated into messenger RNA, and which the messenger RNA is going to be used to it will be translated into what we call a polypeptide chain when it goes in the cytoplasm. So that flow of information, as you can see it from here, that is what they call the central dogma. As we move from this DNA to RNA, and from RNA to proteins, that is what they usually call the central dogma. So as when we are moving from DNA to RNA, that is transcription. When we are moving from RNA to protein, that is translation. Now, we can also have, in preparation for protein synthesis, we can also have duplication of those genes which are in context. So we can also have DNA replication. So central dogma simply talks about how information is flowing from DNA, the information that is encoded within the DNA, how it is going to be converted into messenger RNA, and then the messenger RNA is going to be converted into a protein. Messenger RNA sequence will be converted into a protein finally. It was suggested by Crick in 1958. That is what he suggested. However, when you look at the concept of the central dogma, is somewhat abused in some organisms, like when you look at the retroviruses. The retroviruses, they go against the central dogma. So they are exceptions of the central dogma in a way that for them, they do not follow the specific chronological order of events, of how information is flowing. For them, they do what they call reverse transcription. When you look at the when we looked at the central dogma, we said that we move information from DNA. From DNA, we go to RNA. From RNA, we finally go to protein. For them, since for them these retroviruses do not have DNA in them, they make copies of DNA using their messenger RNA in the presence of an enzyme known as reverse transcriptase. So since now we are moving from RNA back to DNA, and from DNA again back to RNA, and then finally go back to making a protein. That is an exception to the normal flow of information, which is supposed to be from DNA to RNA, from RNA to protein. So in reverse transcription, we are having copies of RNA which are going to be used as templates to make DNA copies in the presence of an enzyme known as reverse transcriptase enzyme. So when we are doing so, then what we are doing is reverse transcription, and it is an exception from the normal flow of information. So closely related retroviruses are known as the retrosporsomes. So usually I've told about the exception of the central dogma. So that is the exception of the central dogma. So we can go ahead and now look at the RNA world. So when you look at the RNA molecules are the ones which are usually they play a very crucial role when it comes to transcribing information, converting this information from the DNA into a form that can be understood in the cytoplasm. So that is what they play. So they play a very central role in protein synthesis. They go again and get the information that is required for synthesizing a certain protein, and then they then they can take it at the ribosomes and use it for synthesizing the different kinds of proteins required by the body. So some of the RNAs can even act as enzymes. You have heard about ribozymes. So ribozymes are very important because sometimes they can catalyze certain chemical reactions. Apart from just being being transducers or carriers of information or adapters for information, they can as well also be enzymes. The RNA molecules can also be enzymes. So when you look at the different kinds of RNA molecules that are used during the process of protein synthesis, you will appreciate with me that we have 3 different kinds of RNA molecules. And these RNA molecules range from messenger RNA molecule. The messenger RNA molecule, it's usually, in fact, it is said that it is the biggest among all the 3 different types of RNA molecules. So it is the biggest, and it is usually a copy of the gene that is being expressed. It is a copy of the gene that is being expressed. As we shall see, you realize that it is complementary to the template strand of DNA. And it is usually, its nucleotide bases are in groups of 3s. And those groups of 3s are known as the codons, which code for an individual amino acid in the protein. So it is usually a copy of the gene that is going to be expressed, and the nucleotide bases, the RNA nucleotide bases, which are arranged on the messenger RNA, are in 3s, which we call triplets, and they are called codons. And each of those codons is going to go ahead and be used to assemble a specific amino acid in the sequence. Now we go ahead and look at we also have the ribosomal RNAs. The ribosomal RNAs, they are the most numerous of all the 3. The messenger RNA, they are the largest among them. The ribosomal RNA, they are the most numerous. There are 4 different types of, you will realize that with the ribosomal RNA is usually made up of 4 different RNA molecules that make up the structure of the ribosome or of the ribosome. So it is made up of RNA molecules and proteins. So about 4 different kinds of, about 4 of them, 4 different kinds of messenger RNAs. So 4 different types of ribosomal RNAs come together in the presence of other proteins, and they make what we call the ribosomal RNA. We shall look at the structure of the ribosomal RNA. So they perform an actual role. What they perform is simply because they catalyze the addition of. So for them, their job is to catalyze. For them, their job usually they do catalysis. When you look at the ribosomal RNA, I've told you about them. They perform the actual catalysis of addition of amino acids to the growing peptide chain. And then finally we have the transfer RNAs, which are considered to be the smallest RNAs. They are smallest molecules, and they act as adapters between the codons of the messenger RNA and the amino acids. We shall look at it in details, their structure. We can go ahead and look at some differences. When you look at the RNA and the DNA, how they are different in certain structures, you realize that the RNA contains the sugar that it contains ribose sugar, and then DNA contains a deoxyribose sugar. This means that at carbon atom number when you look at the ribose sugar has a hydroxyl group projecting outwards from carbon atom number 2. But when you look at DNA, because it is deoxyribose, it lacks one of the oxygens it lacks an oxygen atom on the carbon atom number 2. So it just has a hydrogen. That is why they call it deoxyribose. Then the RNA contains the base uracil. The DNA contains thymine instead. RNA is usually single-stranded and DNA is usually double-stranded, with some exceptions of some bacteria. RNA is usually short, one gene long at most, and when you look at DNA, it is usually large, made up of very many different genes all put together. So this is another illustration to show the meaning of what we call a central dogma. Remember, always central dogma, transcription, translation. We have replication, DNA replication, transcription, translation. Exception is reverse transcription, with the exception of reverse transcription in retroviruses. So we can go ahead and now look at, having looked at the central dogma, we can go ahead and look at the whole process of transcription. Now when you look at transcription, transcription is divided into about 3 different stages. But as we start, we need to go ahead and look at the step by step. It all begins with what we call initiation. But before initiation happens, what we need to know is when you look at the transcriptional unit of a DNA, a transcriptional unit of DNA is usually comprised of about 3 different components, and I want to talk about each of them before we can go ahead and look at the whole process. So the transcriptional unit of DNA must be made up of a promoter, and these promoter regions, like the way you can see them here, we have promoter regions which are located upstream on the 5' end of the DNA. So promoter regions are located on the 5' end of the DNA, that is to say upstream. The DNA is usually, so it is a DNA sequence that provides binding site for the RNA polymerase. So that is where the RNA polymerases can bind, and then they start transcription process. So they are known as promoter regions, and they are located 5' upstream. Very important for the attachment of DNA polymerase. So these DNA polymerases usually attach to specific promoter regions. You can see that in eukaryotic gene we have the promoter region at negative 55. Negative 55 means that it is located 55 nucleotides upstream, up from the start. Then we also have the negative 10, which is known as the tartar box. Then we have the negative, we have also the positive 1. So those are the different promoter regions that we have in our transcriptional unit. That is where the RNA polymerase will go ahead and bind, and then it will initiate the process of transcription. Now, on top of that, we usually have, within the transcriptional unit, we usually have structure genes. These structure genes are the ones which, they are the 2 strands of DNA which have opposite polarity. That is to say, one of them, when you look at DNA-dependent RNA polymerase, what it does is it catalyzes polymerization in a 5' to 3' direction. So as it is catalyzing the polymerization of in 5' to 3' direction, so the DNA strand having a polarity of 3' to 5' direction, polarity acts as a template strand. So as we are synthesizing our RNA strand in a 5' to 3' direction, what happens is the DNA, when you look at the DNA strand which has a polarity of 3' to 5' direction, is going to act as the template. Remember, within our structure gene we have the 2 strands of DNA, but of those 2 strands of DNA, one is chosen to be the template, and the one that is chosen to be the template is the DNA strand that is read in the 3' to 5' direction. As it is being read, the RNA polymerase is going to synthesize new RNA strand in a 5' to 3' direction. So the other strand known as, of the DNA having the 5' to 3' polarity is complemented to the template strand, and the sequence of DNA in this strand is usually the same as the one for RNA, except at a location where we usually find thymine, which is usually replaced with uracil in the RNA. So this is actually the coding strand of the DNA, and the information on this strand of DNA is usually copied in the RNA, and that is why this strand is known as the sense strand. So on top of that, in this transcriptional unit, the other component that we usually have that is very important, we usually have the terminator. The terminator region, or the terminator is the one that is usually found, for it is found downstream at the 3' end of the DNA. So it defines the end of transcription, and when we reach that point, we shall see the process of termination of the whole process of DNA. So what usually happens, remember that after binding of the RNA polymerase to the promoter region, what happens next? It causes local unwinding of the DNA duplex. So when it causes local unwinding of the DNA duplex into two chains of the regions, now it exposes a certain sequence of DNA molecule, a certain sequence of nucleotides known as ATCG bases. So they project outwards into the nucleoplasm. Only one strand functions as a template, and that is what we call the antisense strand. So what usually happens, and the other one which is usually complemented to the actual coding, is usually the actual coding strand or the sense strand. So the ribonucleoside triphosphate joins the bases of the so what usually happens, we usually see now growing the chain. So that is the whole process of initiation of the process of transcription. That is how initiation happens. We are going to look at how it is different in prokaryotes and how it is different in eukaryotes. But for now, that is what we are looking at. So we have looked at this is eukaryotic, not prokaryotic. We are going to see the differences that exist between the two in each of those stages. So having looked at that, now we can go ahead and look at the next step, which is elongation. So when you look at the whole issue, as transcription process usually proceeds, the hybrid DNA and RNA molecule dissociates and makes RNA molecules free. So what usually happens, we continue, but remember, as we are synthesizing our DNA, we are synthesizing our RNA, we are synthesizing RNA in the 5' to 3' direction. And the one that is acting as a template strand is the one that is the DNA strand that is in the 3' to 5' direction. So that is all. So finally, when that whole process is done and we have transcribed the entire gene of that specific, when we have finally transcribed the whole gene that we need to make that specific protein, what follows thereafter, we usually, when the RNA molecule reaches what we call the terminator signal of the DNA, it leaves the DNA and fully formed RNA molecules are released. So this process of termination of DNA, it comes with very many different kinds. You'll find that in some books they will tell you that we have what we call the row-dependent RNA row-dependent termination, then the row-independent termination. So there are different kinds of termination. But what you need to remember is when the RNA polymerase reaches that terminator signal on the DNA, that it is like telling it that this is the end of the structure unit of this gene. So what happens is it drops off, and as the RNA grows, when it drops off, the RNA is released. Okay, so as we are going to see the differences, so we are going to go ahead and see the differences that exist between eukaryotic transcription and prokaryotic transcription. How different are they? That is what we are going to go. So when you look at what are the differences that exist between transcription in eukaryotic cells and then translation. When you look at in transcription eukaryotic cells, we usually have multiple RNA polymerases. As we shall see, the multiple RNA polymerases, we shall look at them, each of them. We usually have, when you look at the prokaryotic, it is only one RNA polymerase that is doing everything. Now, we have 5' capping in eukaryotic transcription. Yet in prokaryotic, we do not have capping. We have splicing. The other side, we don't have splicing. We have a poly A tail being added. In other words, we have what we call post-transcriptional processing of the heteronuclease RNA, which is not existing in the prokaryotic DNA or in the prokaryotes. Now, when you look at the different multiple polymerases, you usually have, in eukaryotes, there are about 3 different types of RNA polymerase. When you look at RNA polymerase 1, it transcribes for rRNA. When you look at RNA polymerase 2, it transcribes for messenger RNA, which is usually known as the primary transcript. When you look at, and also some small heteronuclear RNA. When you look at RNA polymerase 3, it is responsible for the transcription of transfer RNA and some small nuclear RNA. Now, when you look at the whole process, when you look at this specific transcription unit of the gene, you realize that there are 2 terms that we usually use simultaneously that help us to describe this transcriptional unit and the gene. You must have heard about a term known as a cistron. A cistron is a segment of DNA that is coding for a polypeptide. That specific sequence or that specific segment of DNA that codes for a specific polypeptide is known as a cistron. And a single structure gene in a transcriptional unit, what we usually call a single structure gene in a transcriptional unit, is known as monocistronic, as the case of the prokaryotic gene. Whereas when you have several different genes having a set of various structure units in one transcriptional unit, then that's what we call polycistronic. So like in the eukaryotic DNA, when you look at it, its gene, the DNA gene, it has a set of different structure genes within the same transcription unit. That is why they call it polycistronic. Monocistronic, we have only one structure. Gene. That means that only one protein is produced at a time. But when you look at for eukaryotic DNA, we have polycistronic, very many different structural genes within the same transcriptional unit. So that means that we can form very many different proteins. So now we can go ahead and, having looked at that, allow me to go ahead and we go through what they call processing of the DNA. So when we are doing processing of the heteronuclear RNA, it is not yet mature RNA, it is nascent RNA. It is still young. When you look at it, you realize that it has both introns and exons. So yet we only need exons. Introns are intruders within the DNA, within the RNA, and they are not coding for anything. So we have to slice them out. And when you look at the exons, they are the ones which are important. So we need to put all them together so that we can use them to translate them into a protein. So now, when you look at post-transcriptional processing of this heteronuclear RNA, there are a number of things that happen. So we can start with what we call capping. So in capping, what we usually do in capping is methylated guanosine triphosphate is usually added at the 5' end of this heteronuclear RNA. And now that is capping. That is what we call a capping process. So you can clearly see that we have added on a cap at the 5' end of the RNA. So you can see it was originally like this. So now you have added on a 5' cap. And I've told you the 5' cap is made up of it is made up of methyl group, it is made up of guanosine triphosphate. So a methylated guanosine triphosphate is usually added at the 5' end of this heteronuclear RNA. Now at the tail end of this RNA, we usually we also do some tailing and modify there. So we usually do what we call polyadenylation. So we usually do polyadenylation. We usually add a polya tail at the 3' end. It is now fully processed heteronuclear RNA. Now, after doing that, we go ahead and now the next step we're going to do is splicing. So during splicing, in eukaryotes, usually from RNA transcription, the DNA is called a primary transcript, and I've told you about it already. So what we usually do in splicing is it happens in the presence of a complex of proteins known as spliceosomes. So the spliceosomes are going to act. It's a complex formed by about 2 enzymes, which are going to go ahead and cut. After cutting, then we bind together. So we have an element of lysis, and then we have an element so after lysing, then we can join back ligase. So ligase enzymes are part, and then we also have lyase enzymes being part of the whole process. So what happens is, specifically, we usually do splicing comes from the 5 small nuclear RNPs and RNAs, which are usually denoted as U1, U2, and U4, U5, and U6, which contain a sequence of which is complemented to the splice junction. So what usually happens here in splicing, we usually remove the non-functional introns. These introns are usually spliced out, and then we remain with the exons. So you can see it here, the splicing process. I've told you the spliceosome is usually composed of different small nuclear RNPs. So what happens is, it comes and attaches, then slices out, it cuts and cleaves. Then after cutting and cleaving, that is to say, RNA polymerase 1. In fact, RNA polymerase 1 is the one that does the cutting. So after it is done with the cutting, then we can have lyase enzymes, which are going to come ahead and join. They will come and join the exons together. So after that is done, now what we usually do, there is also a phenomenon known as alternative RNA processing or splicing, alternative splicing, where we can, for instance, if when we want to form different kinds of the same protein, take an example, we want to form different kinds of antibodies like IgG, IgA, IgM. What we usually do is we just try to alter the arrangement of these exons. For instance, if we had exon 1, exon 2, exon 3, exon 4 in that order from 1, 2, 3, and 4. So we can do splicing in a way that we can cut and then we change the order to 1, 3, 2, and then 4. We have changed the order so that we can form a different protein, maybe IgM. Then we can go ahead and splice again 1, 4, 3, 2. That's a different order. That is alternate splicing, and it enables us to form different proteins of the same kind, like when we are forming antibodies. That's what we usually do. So basically, in this video, we have been looking at the central dogma of DNA, central dogma, where we have been looking at information traveling from DNA to RNA, and that is transcription. From RNA to proteins, that is translation. And then we went ahead and looked at the process of transcription, where we said in transcription is made up of basically 3 stages. We have initiation, which exists at the promoter region, and we said the promoter regions are usually made up of, they are usually found at the 5' end upstream. What do I mean? Upstream, I mean at the 5' end of the DNA. We have the negative 35, we have the negative 10, which is the tart box, then we have the positive 1. So it means that that 35, it is 35 nucleotides up the stream where we are going to start from. We also looked at the DNA polymerases, and we said that in eukaryotic transcription there are about 3 of them. The first one, DNA polymerase 1, which is very important for rRNA, polymerase 2, very important for mRNA and heteronuclear RNA. We also said that polymerase 3 is very important for formation of tRNA, which is the smallest among all those RNA molecules. We said a transcriptional unit is made up of both. It is made up of 3 components. It is made up of a promoter. It is made up of the structure gene. The structure gene can either be polycistronic or monocistronic. We said in a prokaryotic cells they are monocistronic. The structure gene is made up of only, when you look at the monocistronic, when you a single gene is made up of only one one functional gene, so one protein. Polycystonic, more than one gene is found within the structure unit. So we can form very many different proteins from that specific structure unit. Then we say the third part, or the third component of the unit is known as the termination signal, upon which, when we reach the termination signal, the RNA transcribe can go ahead and be released. The RNA polymerase breaks off from the DNA, and then the RNA transcribe is released into the cytoplasm. Thank you so much for listening. I remain teacher Dr. Mgomba Ronald. If you are it is your very first time to be on the channel, consider, if you've enjoyed and you're really pleased, you can go ahead and subscribe to our YouTube channel. You can consider to share with your colleagues so that you do not be selfish with knowledge. You can consider to like. You can consider to even ring the bell icon so that you are always notified about the new videos that we have made. I remain teacher Dr. Mgomba Ronald. Till next time.